Right. So
We can.
great. Yes, as Nantia said, I'm Nick Quinn-Bohmann. I'm a postdoc in the Gibbons Lab at ISB, an Institute for Systems Biology in Seattle, Washington. At the Gibbons Lab, we focus on developing computational and experimental approaches for exploring the gut microbiome, and then using that learning to understand and engineer host microbe systems. Today, I'll be talking a little bit about a project of mine that has been going on for several years that focuses on exactly this, wherein we use community scale metabolic modeling to predict the individual responses to different microbiome-targeted therapeutics. Without further ado, let's jump into that. So as most of you probably know, our body and all of the surfaces in our body that are exposed to the outside world are colonized by trillions of microbes. So your skin, your upper respiratory tract, your gastrointestinal tract are all colonized by these microbes. And it actually wouldn't be that much of a stretch to say that our bodies are actually more microbial than human, in a way of speaking. If we look at cell counts between our own endogenous cells and these microbial cells, we see that it's about even between the two. Obviously, the microbial cells are 100 to 1,000 times smaller, so they end up only making up 1 or 2 kilograms of mass, but they are there, and they are very important. And in fact, if we look at unique genes between our microbiome and us and our genome, we can see that the microbiome sort of metagenome outnumbers our genes by a factor of nearly 100. And it turns out that many of those genes are actually critical for our own health and physiology. Throughout the course of human evolution, we've sort of outsourced some functions that our body does not do to these microbes that live in and on our body. And just to name a few different functions, the microbiome helps us with digestion of certain dietary components. It helps to regulate our immune system and train the adaptive immune system to recognize potential pathogenic invaders. And it also helps to regulate our metabolic health through a number of different functions, but primarily through the production of microbially derived metabolites. So in our lab, we primarily focus on what's called the gut microbiome, which is the most dense collection of microbes that live in our body, in our lower gastrointestinal tract, in the mucus lining of the colon. These bacteria are critical for our health, and they help us to do a number of different things. Primarily, they help us with digestion of certain dietary components that our bodies aren't adept at digesting. One of the primary ways that the microbiome, the gut microbiome in this case, impacts our health is through the production of these small molecules or microbially derived metabolites. And some of these metabolites are good for us, and some are rather detrimental. So for instance, if you're eating a diet of fiber-rich food, maybe you have a lot of vegetables and legumes, you're likely promoting the production of what are called short-chain fatty acids, or SCFAs, by the microbiome. These metabolites then get transferred through the colonic epithelium and into our body. And there they serve a number of beneficial functions. So they help to maintain the integrity of the gut barrier, keep the lining of the colon healthy. They regulate metabolism through the production of a number of different hormones. And it's been found that they can also ameliorate inflammation and insulin resistance and potentially even lower blood pressure. So overall, this is a very beneficial class of metabolites. It's worth noting, too, that over several generations, the amount of fiber that's in the average person's diet has, specifically in Europe and North America, has decreased. And as such, our microbiomes are less adept at producing these SCFAs, which can contribute to some of these chronic diseases that we see are rampant in society. So things like certain cancers, diabetes, and obesity. Conversely, certain metabolites that the microbiome produces are also fairly detrimental to our health. So a diet that's rich in things like carnitine and choline, which are found in eggs and red meat, that can lead to the production of trimethylamine, or TMA, which after moving into the body is converted into TMAO, trimethylamine N oxide. This is a well-known risk factor for several cardiometabolic diseases. It's shown to increase insulin resistance, cardiovascular risk, and also systemic inflammation. So really what this shows us is that the microbiome is sort of this little metabolic factory that takes what we give it in terms of dietary inputs and converts that into outputs, which can either be helpful or detrimental to us. And that's sort of the paradigm that we'll want to focus on for the rest of this talk. Before we really dive into that, I do want to key in a little bit more on short-chain fatty acids, or SCFAs, since those are the sort of the main character of today's talk. They're the class of metabolites that we'll focus on today. SCFAs are fatty acids with between 2 and 6 carbons on their carbon chain, with acetate, propionate, and butyrate being the most commonly produced. They're primarily produced from the microbiome, from the fermentation of dietary fiber, like I mentioned, and they serve a number of different physiological functions. So they help to regulate our metabolism through the production of GLP-1 and PYY, which regulate blood glucose and our appetite. Propionate has been found to regulate serum cholesterol and help to regulate hepatic lipogenesis, which can prevent what's called fatty liver disease. And it's also been found that all 3 of these metabolites are anti-inflammatory, primarily through the inhibition of cytokine production, so TNF-alpha and IL-6, among other cytokines, as well as, in the case of butyrate, HDAC inhibition, which leads to increased proliferation of T regulatory cells, which shift our immune system into sort of a anti-inflammatory state. So sort of at a system scale, what these SCFAs do is they really impact health and well-being in a number of different areas in our body. So through the gut-brain axis, they help to increase neurogenesis and help to maintain the blood-brain barrier. They also help to regulate hepatic lipogenesis, like I mentioned, and they can help to ameliorate insulin resistance. Locally in the colon, they're very important, as SCFAs act as the primary energy source for the colonic epithelial cells that line our gut, and they help to maintain the gut barrier function, so keeping those tight junctions tight. And as I mentioned, they're also protective against cardiovascular disease and strongly anti-inflammatory. So overall, SCFAs are very beneficial. It's one of the most important things that our microbiome does for us is producing these metabolites. And obviously, there seems to be a lot of benefit to increasing the production of SCFAs from the microbiome. So how can we do that, right? That's sort of the central thesis of the first part of this talk today is, can we engineer the microbiome to increase SCFA production? And on its face, this seems like a very easy problem to solve. You just chuck a little bit more fiber in someone's diet, and presumably they would start producing more acetate and butyrate and propionate. However, it turns out that this is actually a slightly more difficult question to answer. And that's because of the great heterogeneity that exists between your microbiome and anyone else's. So as you probably know, your microbiome is very unique to yourself, sort of like a fingerprint. And the composition of your microbiome is definitely will look different from someone else's. The functional outcome of that, meaning that the metabolic capacity of your microbiome will differ from anyone else's. So that given the exact same dietary input, the metabolic outputs will likely look different. As we can see on the right-hand side here, this was an experiment in which participants were given the exact same dietary input, and then short-chain fatty acid production was measured, and we can see that there were drastically different levels of each of the 3 SCFAs being produced by these individuals. The reason for this is, like I mentioned, differences in metabolic capacity, as well as differences in taxon-taxon interactions. So essentially with different taxa being present in your microbiome, you might get some cross-feeding interactions that occur in one sample, but not in another sample, where a given species is consuming a dietary component, producing a metabolite, which is then consumed by a second species, and so on. And so everyone's microbiome looks a little bit different. And if we combine that with the fact that adding fiber to your microbiome in a scenario wherein your microbiome is not able to actually degrade it and is unable to digest that fiber, this can actually lead to increased inflammation, since that fiber is just sort of sitting there in your gut, and this can lead to increased methanogenesis and ultimately a more detrimental outcome than if you hadn't added the fiber in the first place. So at a high level, what this means is that a beneficial diet for one person might be detrimental, highly detrimental for another, which kind of goes against the classical paradigm of how we've looked at nutrition in the past, is that there should be such a thing as a sort of healthy diet and an unhealthy diet. But it turns out that might not be the case. And what we might be interested in is using predictive modeling to predict exactly which diets would be helpful for one person, but not necessarily for another person. And so, yeah, this is sort of at a high level what we're thinking about here. If we think of, again, the microbiome as being the sort of metabolic factory that takes in its inputs in the form of diet, and it has sort of its unique machinery based on which microbes live in a given microbiome. How can we engineer it such that the outputs, so the microbially produced metabolites, are beneficial to us rather than detrimental? And the two tools that are at our disposal to do that are, one, prebiotics. So this is sort of supplements that we can add to a diet that contain dietary fibers or other beneficial metabolites that could improve the sort of input stage of our little metabolic factory, as well as probiotics, which are live biotherapeutics, or sort of putatively considered healthy taxa that we can add to the microbiome to sort of change that the machinery that exists in the factory, such that the outcomes are more favorable for us. So that's generally what we're after here. But how can we do that? I've mentioned several times about predictive modeling, and there might be a few different ways that you would conceptualize this. But what we use is a tool called flux balance analysis. Now, I imagine several of you are probably familiar with flux balance analysis, or FBA, but I'll go through it here just in case. So FBA is a mathematical framework that allows us to recapitulate the metabolic capacity of a microbial system, be it a single taxon or a community. And using objective function optimization, so essentially identifying a biological goal of that system, for instance, the production of biomass, we can then find the set of fluxes through that metabolic reaction network that will maximize that objective. So this all starts with creating a metabolic reaction network, like you see on the top left here. We can usually get this from an annotated genome of a microbe, where we can sort of infer through the genome what enzymes are present, and from there we can figure out what reactions are taking place in this microbe of interest. We can represent this reaction network mathematically by converting it to a matrix, wherein all of the rows are metabolites, all of the columns are reactions, and then the matrix shows the stoichiometric coefficient of each metabolite within that reaction. This then sort of creates our baseline model. But in order to find a solution that satisfies our constraints, we do have to set our constraints to begin with. The first constraint that we take or that we apply in flux balance analysis is the steady state constraint. So essentially, we're assuming and we're solving for a solution in which all of the input fluxes are balanced by the output fluxes, and there's no accumulation essentially of any metabolites within the system. We also then add individual bounds on each of the reactions, given prior information about those reactions. So for instance, if we know that a reaction thermodynamically can only happen in one direction, we can set that as a bound. Alternatively, if we know that there's only a limited amount of a substrate to be imported by the system, we can also set that as a bound and prevent unlimited import of that metabolite into the system. This constrains our model such that we have a defined solution space, which you see in this second panel here, within which all of the points within that constrained solution space satisfy our constraints. To find a unique solution within that solution space, we then return to this an idea of an objective function, which in the case of a single microbe, we primarily use biomass production. And we very simply just find the set of fluxes in our allowable solution space that maximizes biomass production in this case. It turns out this works really well for modeling single microbes and being able to sort of recapitulate their metabolic behavior in a system. What we want to do, however, is model a whole community of microbes. So the whole gut microbiome, which is taxonomically very rich. And to do that, we actually find that we run into trouble if we use this same approach with biomass production being our sort of objective that we're optimizing for. And here's just an illustration to show you why that's an issue. So consider this very, very simple community wherein we have two taxa that are functionally identical, and they're both present in sort of 50% relative abundance, so equal amounts. And we first try to just optimize the system for maximal biomass production. What we find is that there's actually an infinite number of solutions that do satisfy that optimal biomass production constraint. So essentially anywhere on this red line satisfies that maximal community biomass production. Now we know that if these are two identical taxa, they should both be able to grow in about the same growth rate and produce the same amount of biomass. So we kind of intuitively know that the terminal solutions, wherein only one taxon is growing at the expense of the other, those aren't very likely to actually occur biologically. And so in order to find a solution to our community growth rate problem, a former lab member of the Gibbons lab, Dr. Christian Diener, developed what's called cooperative trade-off flux balance analysis, wherein we first determine what that optimal maximal biomass community growth rate is. And then we add a second step, an L2 regularization step, wherein we minimize the sum of the squares of the individual growth rates, so the growth rates of each community member, and find an optimal solution there. That will still exist on this red line, but this returns to us a unique solution that balances growth more evenly between community members. However, we do sometimes run into issues where that maximal community growth rate, the one that actually achieves sort of the maximal biomass production, that's only achievable when one or two taxa are growing at the expense of all the other taxa in the community. And this violates our first assumption, which was that if a taxon is measured to be present in the system, it should be able to grow. Because if it wasn't growing in the environment of the gut microbiome, it would simply be diluted out into extinction, and we wouldn't have measured it there in the first place. So to overcome that, we introduce what's called a trade-off parameter, wherein the L2 regularization step is done at a predetermined fraction of the maximum community growth rate that we calculated in the first step. So essentially, we take a fraction, usually somewhere between 0.6 and 0.9, and we essentially find the cooperative L2 regularization set of fluxes, or a set of growth rates, I should say, at that level. Overall, what we find is that this works pretty well. So that's sort of the question, right? Is if we introduce this extra step, do we end up with biologically feasible growth rates? And first, we can see that the distribution of growth rates seems to look a lot more reasonable. So here we can see on the x-axis growth rates of individual species, and then sort of the distribution of those growth rates within a community at various levels of this trade-off parameter. And what we'll notice first is that if we ignore the L2 regularization step entirely, we have a very small number of taxa that are growing with a high growth rate, so above 1, and then almost everything else is growing at a super low growth rate of like 10 to the negative 16th, which, for all intents and purposes, is no growth. That's the growth rate of 0. Since again, if you're growing too slowly in the microbiome, you will be flushed out. If we include the trade-off parameter, but keep it high, and we see there is a shift to the right, and some things are growing, but we still have a high number of taxa that are growing at a very, very low growth rate, so essentially not growing at all. But once we see that we're in a regime around 0.7 and below for that trade-off parameter, we do see that most taxa are able to grow, which matches our intuition that all the taxa we measure should be able to grow. And it turns out we can also experimentally validate this. So there was a paper several years ago that came out from the Scale lab where they showed that bacterial replication rates can be inferred from coverage profiles from metagenomic sequencing. So essentially, since bacterial genomes replicate from the same origin of replication, if you map this coverage profile and map the number of reads that correlate to each part of that bacterial genome, you can actually infer the replication rate sort of from the slope of this coverage profile. And if we compare those inferred replication rates against predicted growth rates from the CT-FBA method, we find that, again, at the trade-off values below 0.7 or so, there is actually significant agreement between inferred replication rates and our predicted growth rates. Obviously, it's not perfect, but there's definitely noise in this in these inferred replication rates. And there's also these models were built without any information about the dietary inputs put into the microbiome. And so those were assumed inputs. And so obviously there will be some divergence between model predicted growth rates and actual growth rates. But this was an encouraging sign that CT-FBA and this MCMM approach, this microbial community scale metabolic model approach, does seem to work rather well at capturing growth rates. So sort of high level again, what this MCMM, so microbial community scale metabolic modeling approach allows us to do is to take metagenomic sequencing or 16S sequencing and use that to construct a community scale metabolic model, recapitulating the metabolic behavior or metabolic capacity, I should say, of the whole microbial community. We can apply to that a diet that essentially limits the metabolic imports into that community that it can be used for growth. And then using the CT-FBA approach, we can then return growth rates for each taxon, as well as metabolic exchanges into and out of each taxon and the community as a whole. This allows us to take a mechanistic look at what's happening within the microbiome, understand who's producing what, how different taxa interact with each other, what codependencies exist, and importantly for the work that we've been doing, examine the efficacy of different interventions, whether they be dietary or probiotic. So to start off, we ask the question of, can we use this method to predict predict short-chain fatty acid production from the microbiome? That's what I'll show you now. So to do this, we actually turn to 4 independent data sets where microbiome samples were collected directly from participants, but then cultured ex vivo. So they were cultured in an anaerobic chamber, and SCFA production was quantified. We did this because SCFAs produced in the microbiome in vivo are very quickly consumed by the colonic epithelium. Like I mentioned, that's sort of the colonic epithelium's primary growth substrate is SCFA. And so it's very difficult to make accurate measures of SCFA production in vivo. And so we did this ex vivo such that there was no interference from the host. We can then compare the SCFA production that was measured from these ex vivo cultures against our MCMM simulation. So simulating the growth of these models and comparing that against actual produced values of butyrate, acetate, and propionate. And what we were hoping for was a very strong positive correlation, of course, between measured values and predicted values. And what we found was that across the 4 data sets, for both butyrate and propionate, we were very well powered to make these predictions. We saw a very strong positive correlation existing between predicted values of butyrate and propionate and measured values of butyrate and propionate. Acetate, interestingly, was not as well predicted. This is likely due to the fact that acetate in many microbial systems acts as sort of an overflow metabolite. So excess carbons are shuttled into acetate production, which means that it's not necessarily a growth-associated process. So the acetate production is not necessarily something that these microbes need directly to grow. And since our optimization function sort of relies on an objective function capturing biomass production and microbial growth, likely that process wouldn't be as well captured as butyrate or propionate production. We can also see this spread out across the 4 independent studies. It's worth noting that each of these studies also included samples that were given just a PBS control, as well as samples that were treated with a dietary prebiotic. So the dietary fibers pectin, inulin, or FOS, which is fructooligosaccharides. And in general, you see that samples that were given these prebiotic fibers did produce higher levels of SCFAs. And our model did a fairly good job of discriminating between high and low producers of both butyrate and propionate, and accounting for the addition of prebiotic fibers. So that was sort of our primary push towards validating that our models are able to predict SCFA production, particularly with regard to butyrate and propionate, less so with acetate. And then our next question was sort of, does this actually tell us anything about the physiological state of the host? It's great to predict this, but is there any clinical relevance to these predictions? So for this, we turned to another cohort of about 3,000 individuals that were in a former precision wellness company called Aerovale. And for each of these individuals, we again took the 16S sequencing and built metabolic models for, representing their specific microbiome. And then we compared predicted levels of butyrate and propionate that were predicted from these models against a set of blood-based clinical labs and chemistries, many of which were markers for cardiometabolic diseases. Knowing that SCFAs act on a number of different bodily functions, but are widely considered to be protective against cardiometabolic diseases. And what we found was very encouraging and interesting. Butyrate, it seems, so butyrate predicted by the model was strongly negatively correlated with a number of these markers for cardiometabolic health. Just to name a few, arachidonic acid is exacerbated generally, and when it exacerbates inflammation at high levels. And we can see that there's a strong negative correlation occurring between model predicted butyrate and arachidonic acid. So high levels of butyrate indicate low levels of arachidonic acid or less inflammation. Similar associations were observed for blood pressure, which is generally associated with inflammatory diseases and cardiovascular disease. C-reactive protein, or CRP, which is a commonly used marker for systemic inflammation. LDL cholesterol, which obviously is highly associated with inflammatory diseases, atherosclerosis, other cardiovascular diseases. LPIR, which is a measure of insulin resistance, and also triglycerides, which are generally increased in incidences of atherosclerosis and other cardiovascular diseases. So this was an exciting result in that it sort of matched our intuition that our model was not only predicting SCFA production, but that those predictions actually had relevance with regard to the cardiometabolic health of the individuals that we were sampling. One interesting note that you might observe in this heat map is that the left column shows measured butyrate in the blood. So for this population, we also had metabolomic measurements, which included serum circulating butyrate. Interestingly, we didn't see any associations between measured butyrate in the blood and any of the blood-based clinical labs. This isn't necessarily surprising since, like I mentioned, most of the butyrate produced by the microbiome is very quickly consumed, and only about 5% of it reaches circulation in an appreciable amount. And even that 5% is very quickly consumed by other parts of the body. And so it's actually very difficult to get accurate readings of serum butyrate in the blood. And really what this shows more than anything is that it underscores the utility of having such a computational approach that allows us to accurately predict SCFA production in a way that doesn't rely on sort of shaky measurements from blood or feces. So given these results, we were sort of interested in seeing how we could use this modeling system to predict how different individuals would respond to simulated interventions, be they dietary, prebiotic, or probiotic. So for this, we used the same cohort of about 3,000 individuals, and we began by simulating the growth of these models on first a European diet. So this is sort of an average European diet taken from a food survey in Austria in 2014, relatively low in dietary fiber. And then we also grew these models using a high fiber diet. So this is a vegan high fiber diet that's rich in resistant starch. This showed generally what we would expect. So given the European diet, we saw very low levels of butyrate production. On the high fiber diet, there was definitely a shift towards higher levels of butyrate production, as we might expect. Interestingly, though, on the high fiber diet, there are still several individuals who are sort of on the left tail of that butyrate production curve. And in that set, we found two separate subsets. So we identified a group of non-responders who essentially just didn't respond very strongly to the switch to the high fiber diet. And we also found another interesting group of regressors whose level of butyrate production actually dropped on the high fiber diet as compared to the European diet. So again, this sort of matches that what I was saying at the beginning of the talk, wherein in a general advice is that a high fiber diet should help, but that might not be the case for every individual. And so what we wanted to do was test a number of different interventions for each of these individuals in the non-responder or regressor group. And so we did just that. We simulated the growth of their specific models using, again, the European and the high fiber diet, but then also each of those diets with a single prebiotic addition, so either the dietary fiber pectin or inulin. And then again, those two diets with the addition of a probiotic species. In this case, we used Faecalibacterium prausnitzii, which is a known butyrate producing species. And the results were pretty interesting, I would argue. There was no single intervention that seemed to be the best for everyone. So again, because of the heterogeneity of the microbiome, we see very many different responses to the same interventions. Interestingly, for some individuals, it seemed like the best intervention, even better than switching to a fully high fiber diet, was simply adding a single prebiotic fiber to the sort of baseline European diet that we were assuming. For others, it seems like it's not even an issue of the dietary substrates that they're consuming, and it more likely is an issue of lacking the functional capacity to actually produce butyrate. And in that case, a compositional shift driven by the addition of a probiotic seems like really the only feasible outcome if the goal is to increase butyrate. So this was sort of our first foray into this idea of tailoring interventions to the individual. And it was rather enlightening in that we saw that there are, there is a very wide range of different responses to both prebiotic and probiotic interventions that doesn't always just match what classical knowledge is, right? So it's not always as easy as just adding a single fiber. And different fibers seem to have different efficacy in different people. Moving forward, what we've focused on more recently is diving in a little bit deeper on the probiotic side. So we have an understanding of how the different prebiotic fibers sort of interact with our microbiome to lead to SCFA production and how that varies between individuals. But the probiotics are an interesting question as well, because they can sort of functionally change that machinery in our microbiome that allows us to produce these valuable metabolites. So for the second part of this talk, I'm going to focus on another related project that was focusing much more on the efficacy of modeling the efficacy of probiotics. So for this case, we used a data set from a controlled probiotic study, wherein a group of individuals was split into a treatment group, which was given a probiotic cocktail containing the 5 probiotic species shown here, as well as an inulin supplement. So both a probiotic and a prebiotic together. There was also a placebo group that it was, of course, just given a placebo without any probiotic or prebiotic in it. And these groups were treated for 12 weeks, and at multiple time points throughout these 12 weeks, the authors did metagenomic sequencing of the microbiome. They measured C-reactive protein and blood glucose, or postprandial blood glucose response in the blood. And then also they conducted qPCR of these 5 probiotic strains just to measure their changes in abundance. At the end of the study, what the authors found was that the treatment group had improved postprandial glucose response as opposed to the placebo group, but there wasn't much of a change in overall C-reactive protein, so a measure of systemic inflammation. It's worth noting that this was only after 12 weeks, and often it could take longer for a probiotic to really take effect. And it was in a fairly small cohort of individuals. But what this represented to us was an opportunity to test whether or not our approach of building microbial community scale metabolic models would allow us to predict whether or not the addition of a probiotic would actually be successful. So would a probiotic given to this treatment group actually be able to grow and take hold in the microbiome and, theoretically engraft? And so to do that, we first turned to the qPCR data. So this was taken across those time points within the study, and it was specific to each of those 5 probiotic strains. What we see is generally 2 of the species, so Akkermansia muciniphila and Bifidobacterium longum, subspecies infantis, they both saw fairly large jumps in abundance between week 0 and week 12, as measured by the inverse cycle threshold value that we're using as a proxy for abundance here. You can clearly see that for both of those, the sort of ending level of abundance is significantly higher than at the start of the study. For the other 3 species, so Anaerobutyricum hali, Clostridium bijurenchii, and Clostridium butyricum, that was not observed. So there was really no jump in enrichment for those, leading us to sort of think there's not as much enrichment or engraftment of those species specifically. Obviously, there are some exceptions, and for some individuals there was higher levels of engraftment than for others, but generally speaking, 2 of the 5 species seemed to successfully engraft, while the other 3 did not. And so what we wanted to see is we wanted to sort of predict the growth rates of these 5 probiotic species using our MCMM approach, and then looking at the growth rates, we can then binarize the growth rates around a threshold. We used 10 to the negative third, which is a fairly conservative growth rate that sort of represents anything below that is essentially no growth. The growth is, again, too slow for it to continually exist in the microbiome without being flushed out. And so our binarization threshold in this case was 10 to the negative third, with anything above that representing growth, anything below that representing no growth. We could then compare that against the results of the qPCR that we just looked at. And what we found was rather encouraging. So in 105 observations, so 5 probiotic species across 21 individuals, we saw agreement in 90, which is about 85% agreement. This leads to a Cohen's kappa, so a sort of a measurement of agreement of 0.7, which indicates substantial agreement. And we can see that some species seem to be better predicted than others. So Clostridium bijurenchii, which generally did not grow very well based on the qPCR data, also did not grow well in the metabolic model context. Anaerobutyricum hali seems to be less well predicted. There is more disagreements in that row than others. This can be better visualized using confusion matrices like we see here. On the top left, you can see sort of the aggregate result across all of the strains, wherein there was 57 instances of no growth occurring and no growth predicted by the metabolic model, and 33 incidences of growth on both fronts, with only 15 disagreements. Across the different probiotic species, you can see that there's similar results. So again, Akkermansia muciniphila and Bifidobacterium longum, species infantis, seem to grow pretty well, which is captured by our model. The 2 Clostridial species were not growing well, which was also captured by the model, and similar for Anaerobutyricum hali. And so this was a pretty exciting result that sort of validates that our metabolic modeling approach is able to predict how well a probiotic species is primed to actually grow in the microbiome that it's being added to. And that can sort of tell us if this is a functionally beneficial thing, or if it'll really have no effect. And of course, we were sort of interested in what the metabolic outcomes of this looked like, right? So if we add these probiotics, what's actually happening to, in this case, SCFA production? That's, again, sort of our primary metabolic endpoint that we're interested in. So to examine that, we modeled we simulated these models in 5 different contexts. So first we took the placebo group, which of course was given no treatment at all. And we saw very low levels of butyrate production for the most part. Next, we took the treatment group, but without either the prebiotic or the probiotic treatment. So just the sort of baseline models. And that looked very similar to the placebo group, as we might expect. Next, we tried using just the probiotic cocktail, so those 5 probiotic species, but without any prebiotic addition to that. This, again, looked very similar to the sort of no treatment group, which makes sense. Without any sort of fibrous substrate to ferment, we wouldn't necessarily expect SCFA production to occur. However, if we add inulin, so our prebiotic, but without the probiotic species, we do see a significant increase in butyrate production. And that makes sense. Our microbiome does have endogenous strains that are producing SCFAs from these fibrous substrates, so that tracks. But what's interesting is that when we add both the probiotic and the prebiotic treatment in concert at the same time, there's again a significant increase over the prebiotic treatment by itself. So essentially what this is saying is that there is a synergy that exists between the prebiotic and the probiotic, where in adding the probiotic makes the prebiotic, the dietary fiber, even more effective, because those probiotic species are able to utilize that substrate more effectively. Interestingly, propionate, on the other hand, another SCFA that was produced by similar means, showed a similar pattern up until that very last context, where we added both the prebiotic inulin and the probiotic, the 5 species cocktail. We actually saw a significant decrease in propionate production. This was puzzling at first, but it actually does make sense if we consider that this probiotic cocktail that we're adding contains a number of butyrate-producing species. So these are species that we expect to actually produce butyrate. And what that's doing then is shifting the entire community away from propionate production and towards butyrate production. So we're essentially shuttling those carbons that we've added via the inulin away from producing more propionate and into producing more butyrate, which is an interesting outcome as well. So finally, and we're running low on time, so I'll try to hurry up a little bit here, but we were interested in sort of exploring how this would look in larger data sets. So we have, instead of just 21 individuals, we've now turned to a cohort of 156 individuals for whom we simulated the addition of these 5 probiotic species in a number of different prebiotic contexts. So essentially we take these 156 samples, we simulate the addition of these 5 species, and then we grow those with the addition of a number of different prebiotics. So inulin, pectin, starch, maltodextrin, hemp seed, which is essentially cellulose, psyllium husk, and then also a complete switch to a high fiber diet. And it kind of just looks like a barcode, but there's actually a lot of interesting patterns that sort of jump out here. So for instance, Akkermansia mucinophila, which is one of our more successful probiotics that we saw, was engrafting in several cases. It seems to really love the high fiber diet. So if you were to switch to a high fiber diet completely, then that would be a very effective probiotic to add. Conversely, anaerobe Butyricum holi seems to only engraft sometimes, but it really doesn't do very well if we do switch to a high fiber diet. There's somehow some metabolic interaction occurring that doesn't allow it to engraft very well at all, especially when you compare that to the results with inulin or psyllium husk. And so really what we're trying to illustrate here is that this methodology can be used to predict the efficacy of not only the prebiotics that we're adding to the community that are producing SCFAs, but also predicting how well these probiotics will engraft. And you can kind of use these in concert to predict the efficacy, of course, of these combined treatments. And yeah, briefly, the SCFA production profile looks very similar to that as well. So in this larger cohort, if we add a dietary fiber like inulin or pectin or starch, we do see that increase in butyrate production as well as propionate production over the sort of no prebiotic context. Interestingly, we see that same pattern again, though, where if we add the probiotic, the 5 species cocktail shown in blue here, we see a boost in butyrate production across the board, but actually a decrease in propionate production across the board. So this is sort of an interesting result, because this is pointing towards the fact that there are these metabolic trade-offs that need to be considered. In this case, this is probably beneficial since we saw that butyrate is highly correlated with many of our clinical markers for improved cardiometabolic outcomes. But really, yeah, we're just trying to illustrate the fact that using these metabolic models, we can predict how well these interventions will work and sort of where the synergies lie between adding the probiotics and then also adding specific prebiotics aimed at making those the probiotic additions more effective. So what is our sort of overall goal from all this? Well, essentially what we're interested in working towards is this idea of a digital twin. So digital twin is essentially a virtual representation of the specific physiology of an individual patient. So you can build digital twins from using deeply phenotyped, longitudinally sampled patient data, anything from genomics to metabolomics to the microbiome metagenomic sequencing that we can integrate into these digital twins. And once constructed, digital twins can then be used to inform the application and the efficacy of interventions. We can essentially computationally test whether these interventions will be successful for a given individual. And our goal is to integrate these sorts of personalized mechanistic models of the microbiome into existing paradigms of digital twins, such that we can, in effect, predict how prebiotic, probiotic, and sort of symbiotic interventions can be successful for a given individual. Long-term, sort of the goal of that, of course, is to replace classical nutrition, sort of this idea of there's a one-size-fits-all sort of healthy diet that works for everyone, which we've seen isn't really the case, with precision nutrition, wherein perhaps the whole diet doesn't need to change, but maybe it's just the fact that a specific prebiotic supplement should be added to an existing diet. Or perhaps your microbiome needs a slight compositional shift to really enable higher levels of SCFA production, for instance, which can be accomplished through the addition of a probiotic. And so we're hoping to build sort of these tailored interventions that result in ideal outcomes for each individual rather than generalizing stuff across the population. So yeah, sort of key takeaways from all this work. SCFA production has been shown to be accurately predicted across diverse dietary contexts and prebiotics. We can see how different prebiotics interact with an individual microbiome and see sort of what are the most effective outcomes. We've also shown that we have a method for reliably estimating the growth of different probiotic species, which we can then use to again, design these tailored microbiome-targeted therapeutics, which goes into my last point. The design of personalized microbiome-targeted interventions can be rationally tailored for maximal impact using this computational approach, combining both prebiotics, so dietary fibers, and probiotics, our live biotherapeutics, aimed at improving outcomes from the microbiome, and in doing so, improving host health through a inflammatory and cardiometabolic lens. So with that, I will thank everyone that's helped me with my work over the last several years. It's been definitely a team effort, and I'm very appreciative of everyone that's helped me along the way. And thank you so much for listening, and I'm happy to take any questions at this time.
Great. Thank you very much, Nick, for your talk. It was very interesting presentation and really nice work done there. So yeah, let's start now with the questions. So if everyone would like to ask Nick a question, please write it in the chat or raise your hand to become an active participating member. Yes, but let me first start. So really nice work, and we got really nice overview how we can use the genome scale metabolic models in a new context, let's say, to engineer the SCFEs, and also related to the cardiovascular diseases. This might be of great interest when of great help when the when those seeing that these models have actually a great effect and great potential in the future and also in clinical application. So how how far are we from this? I mean, what is your opinion on that? Now, you showed us that those models are working very good. You validated the models and you were also able to simulate new outcomes. So, what would be the next outlook for the future? Let's say, and how far we are from the clinics.
Sure. Yeah, it's a great question. I think we're actually closer than people realize. That it's really within the scope of what's already achievable. Microbiome sequencing is fairly cheap at this point. It's fairly inexpensive, and the actual computational power that's required to build these models and run them is also manageable on even a desktop computer. And so I think really the biggest hurdles that are in the way of this is sort of making it marketable to the general public. So making it something that's effectively easy enough for an individual to get their microbiome sequenced, and then turn that into essentially suggestions of prebiotics and probiotics and interventions that could be helpful for an individual. But yeah, I think the methodology exists, and I think there's interest in turning this into a clinical application already. So I think it's fairly close.
Exciting, really nice, great. So we do have a question in the chat. So if you build a personalized, RJ Murray asks, if you build a personalized microbiome profile or model, how useful would it be, if at all? Should the subject undergo something like chemotherapy or severe GI infection that wipes out most or all of their gut biota?
That's a really important point, and a good question. So when the microbiome is wiped out in the case of chemotherapy or GI infection, you end up in a very crucial state where essentially you're very susceptible to pathogenic invasions of bad bacteria, things like Clostridium Clostridoides difficile, C. diff. And while some of the work that I presented here today maybe wouldn't be as helpful, there is ongoing work in our lab that's focused on using metabolic models, personalized microbiome models, to predict the invasion of these pathogenic taxa. So you could use a model like this to predict whether or not you are now in a state where your microbiome is very vulnerable and prone to invasion. We are currently working on models that can predict whether or not your microbiome specifically is at risk of pathogenic invasion or not. The other thing I'll say is that that's often a time where probiotics can be very beneficial, since you want to sort of repopulate your gut to with healthy bacteria, things that'll be essentially pushing out pathogenic invaders and helping to essentially repopulate your gut with things that are helpful. So being able to, again, predict the success of probiotics given the specific context of your microbiome would also be very helpful in that case. So great question.
Thank you. And do you consider adding into the analysis or model reconstruction more layers of data, like proteomics, for example?
Certainly. There's ongoing work that's aimed at looking at sort of transcriptomic data to determine exactly which metabolic reactions are actually taking place. So, right, we have these metabolic models that have essentially metabolic capacity. So what could be happening, but you could integrate in transcriptomic data that's actually telling you which reactions are in fact taking place, not just potentially taking place. So that's one side of it. Another ongoing project that we've been looking at is incorporating sort of an additional model that focuses on the interface between the microbiome and the host. So you could build a metabolic model of sort of host side metabolism of the colonic epithelium, and I mean that would obviously be using proteomic and metabolomic data, as well as genomic data, and then you could think about building sort of this dual model of microbiome and the host, and you could actually model the exchange of metabolites directly. So right now, all we're doing is predicting what metabolites are being produced by the microbiome, but you could take that a step further and say not only what's being produced, but what's actually making it across that barrier and being consumed by the host, and where is that ending up? Is that going into circulation via the portal vein to the rest of the body? Is it being consumed by the colon? Is it these sorts of multi-tissue metabolic models definitely leverage multi-omic data, and that I think is probably the long-term future of this sort of work, is integrating with other body systems.
Yeah, definitely. Good. Maybe another question from my side. So how do you handle because like the microbiome is very dynamic. How do you handle these dynamic phenotypes, like across the time time?
It's a good question. Sorry, I didn't mean to cut you off, but it's an important question. Is sort of the microbiome does shift a bit. We find actually that the general composition of the microbiome is fairly stable from person to person. If you really want to shift the overall composition of the microbiome to make it look substantially different, that takes serious intervention in terms of dietary shifts, so switching your whole diet completely. And even then, it takes on the scale of several, like 6 to 8 months to over a year to see dramatic shifts. Unless, of course, you're talking about the case of GI infections or antibiotics, which are wiping out your gut. But in general, the microbiome is fairly stable. And so that isn't so much of an issue if we can really capture sort of the central metabolism of your microbiome as it stands now. So most likely, unless something drastic happens, look very similar a year from now as well.
So I suppose that in your study cohort, you consider relatively close-aged people, not like because also microbiome changes with age, right? Younger people with older ones. So I assume that you have some similar aged cohort.
Yes, so it definitely does change with age, and we found actually that there's an interesting phenotype that occurs where as you get older, in healthy aging, your microbiome gets more unique from everyone else. Essentially, there's a uniqueness score that can be calculated, and that increases in healthy aging. That being said, though, that the uniqueness of your microbiome is something that's captured in metagenomic sequencing. So even if you have an older person whose microbiome is very distinct from a younger person, that would be something that's captured by the sequencing and then would go into the design of the model. So it's not necessarily something that we consider directly when putting together data sets, since any differences in any compositional differences would already be accounted for in our model construction.